What's up, guys? Welcome to another session with Mr. Abdul Wahab. This is the SAT practice test number 1 again. In the previous two videos, we worked on the first and the second passage of the reading section. Today, we're going to work on the third passage of the reading section, and the passage that we have for today is actually a science passage. So, again, we're going to show that these things do not have to be necessarily tough or hard to understand, that they are actually accessible to any student who actually knows how to go about reading and comprehending this whole thing. So again, questions 22 to 31 are based on the following passage and supplementary material, which is of course this table right over here. As usual, we're going to begin with the intro. So this passage is adapted from J.D. Watson and F.H.C. Crick. Genetical implications of the structure of deoxyribonucleic acid. Deoxyribonucleic acid is basically the long name for DNA. Okay, so which is Watson and Crick deduced the structure of DNA using evidence from Rosalind Franklin and R.J. Gosling's X-ray crystallography diagrams of DNA and from Erwin Chargaff's data on the base composition of DNA. So let's begin immediately. Okay, so the chemical formula of deoxyribonucleic acid, which is also known as DNA, is now well established. So today we know what the chemical formula of DNA is. So the molecule is a very long chain, the backbone of which consists of a regular alternation of sugar and phosphate groups. So this is very important. When you're reading this, you need to focus on tiny details like these. So the molecule, which is basically the DNA molecule, is a very long chain, the backbone of which, the base of which, consists of a regular alternation. When you have an alternation, you have a repeated kind of a pattern, okay, of sugar and phosphate groups. To each sugar is attached a nitrogenous base, which can be of 4 different types. 2 of the possible bases, adenine and guanine, are called purines, and the other 2, which are thymine and cytosine, are pyrimidines. So far as is known, the sequence of bases along the chain is irregular. So we have lots of names here, but then again, there's no need to panic. The whole thing is really organized for us. Again, to each sugar, okay, so again, the DNA, okay, the backbone of which consists of a regular alternation of sugar and phosphate groups, and to each sugar is attached a nitrogenous base, and this nitrogenous base can be of 4 different types. 2 of the possible bases, which are nitrogenous bases, which are adenine and guanine, are called purines, and the 2 other, which are thymine and cytosine, are called pyrimidines. So far as is known, the sequence of bases along the chain is irregular. Now, the monomer unit, I don't know what the word monomer is, now consisting of phosphate, sugar, and base, is known as a nucleotide. So basically, when you combine the phosphate group, the sugar, and the nitrogenous base, you get what is known as a nucleotide, okay? So, again, for the SAT, you really don't need to come to the exam with any previous knowledge of anything, because you're going to answer all of the questions depending on what's stated in the text itself. So the first feature of our structure, which is of biological interest, is that it consists not of 1 chain, but of 2. So there are 2 chains in the DNA. These 2 chains are both coiled around a common fiber axis. Okay, so they're coiled around each other, and this thing that they're coiled or rotated around is a common fiber axis. It has often been assumed that since there was only 1 chain in the chemical formula, there would only be 1 in the structural unit, of course. Okay, so again, in the past, they thought that there was 1 chain in the chemical formula, and so there would be only 1 in the structural unit. However, the density taken with the X-ray evidence, the X-ray crystallography that was mentioned in the introduction, suggests very strongly that there are 2 chains, just as they mentioned right over here. Continuing. The other biologically important feature is the manner in which the 2 chains are held together, the way in which they are coiled together. This is done by hydrogen bonds between the bases, the nitrogenous bases. So these nitrogenous bases are held together by hydrogen bonds. The bases are joined together in pairs. A single base from one chain being hydrogen bonded to a single base from the other. So one nitrogenous base is basically joined together to another nitrogenous base using a hydrogen bond. So this is how the two chains are coiled or connected together. The important point is that only certain pairs of bases, this is important, okay, so the important point, they're saying it's important, so it has to be, is that only certain pairs of bases will fit into the structure. One member of a pair must be a purine and the other a pyrimidine in order to bridge between the two chains. So if a pair consisted of two purines, for example, there would be no room for it. So there would be no space for two purines to basically join together within the coil. We believe that the bases will be present almost entirely in their most probable forms. If this is true, the conditions of forming hydrogen bonds are more restrictive, they're harder. And the only pairs of bases possible are adenine and thymine and guanine and cytosine. So basically adenine, for example, can occur on either chain. So you could have adenine on either one of the two chains, but when it does occur, its partner on the other chain must be thymine. So adenine can only bond with thymine using a hydrogen bond, and guanine can only bind with cytosine together across the two chains. So basically, the phosphate sugar backbone, again I know the word backbone, which is basically the base of the DNA, okay, of our model, okay, is a completely regular, okay, so again, this phosphate sugar backbone of our model is completely regular, repetitive, but any sequence of the pairs of bases can fit into the structure. Basically, since this backbone, this repetition of adenine and thymine or guanine and cytosine, it could be in any kind of a sequence, but you're going to find this pattern throughout the whole DNA. So it follows that in a long molecule, many different permutations are possible. So they could be in any order. So you could have, for example, adenine, thymine, and then another adenine and thymine, and then a cytosine and a guanine. Or, for example, you could have 5 adenine, thymine bonds together, and then 3 cytosine, guanine. So you could have any range or different kind of permutations, but in every case, the adenine must be bonded to a thymine and the guanine to a cytosine. And it therefore seems likely that the precise sequence of bases, okay, is the code which carries the genetical information. So basically this sequence of bases, according to this sequence, this is the code that contains the genetic information. So depending on what sequence they're put in, this is gonna convey different genetic information in the cell, I would say. So if the actual order, okay, of the bases on one of the pair of the chains, okay, so if the actual order of the bases on one of the pair of chains were given, one could write down the exact order, okay? Take a look at this, the exact order of the bases on the other one. So basically, if I give you adenine, you will automatically know that on the other chain you will have thymine, of course. And if I tell you that then there's cytosine, so you already know that on the other chain you have guanine, and so on. So by knowing one of the chains, by knowing one sequence of one chain, we can know the sequence of the other chain to which the first one is bonded, because of the specific pairing which they mentioned before. Thus, one chain is, as it were, the complement of the other, and it is this feature which suggests how the deoxyribonucleic acid molecule might duplicate itself, okay, how it might basically replicate itself or copy itself. The table shows for various organisms the percentage of each of the 4 types of nitrogenous bases in that organism's DNA. Take a look at this table. We're going to get back to it when we go through the questions. I just want to show you, okay, so here we have adenine, guanine, cytosine, and thymine, okay, and as we said before, okay, so the adenine is going to join with the thymine, okay, and then of course the guanine is going to join with the cytosine, okay, so this is how they are paired in a sense. But of course, we could also divide them in another way. So basically, adenine and guanine are the purines. So those two columns, they refer to the purines, of course. Okay, so I'm going to write purines right here in a very terrible handwriting, of course. And then, of course, cytosine and thymine, those are the pyrimidines. I'm going to just write a few letters of it. So just, okay, this is not working. So that's how they are bonded. I care about this, okay, because this was repeatedly mentioned in the text. Now let's go down to the questions. The authors use the word backbone in line 3 and 39 to indicate that only very long chains of DNA can be taken from an organism with a spinal column. The word spinal column wasn't even mentioned in the text, so I'm not going to even bother trying to understand what this sentence is even supposed to mean. The main structure of a chain in a DNA molecule is composed of repeating units. Now, yes, this is true, okay, because again, they talked about the alternating, irregular alternation. Okay, there is repetition, there is a pattern, as we said. And so this actually goes in line with what we've read, okay? So B sounds good. Now, a chain in a DNA molecule consists entirely of phosphate groups or of sugars. Now, you might be tempted to say that this is correct, but actually take a look at the word or, okay? Now, a chain in a DNA molecule does not consist entirely of phosphate groups or sugars. It contains phosphate groups and sugars. So that's why C is wrong. Nitrogenous bases, okay, form the main structural unit of DNA. Well, not really. I mean, it's one of the units as far as I understood it. Again, I'm not a scientist, I'm not a chemistry major, and I don't need to be to answer this SAT, to go through this SAT passage and questions. So B is the best answer. Now, a student claims that nitrogenous bases pair randomly with one another. Which of the following statements in the passage contradicts the student's claim? So a student is claiming that they could pair up randomly with one another. Now, of course, we know he's wrong, okay, because as we said, the adenine needs to pair with the thymine and the guanine with the cytosine according to the passage. So we need to find the evidence that contradicts the student's claim. We have these lines, so let's go 5 to 6. Okay, to each sugar is attached a nitrogenous base, which can be of 4 different types. This doesn't actually negate what the student said. This doesn't contain the negation. So far as is known, the sequence of bases along the chain is irregular. Well, again, this doesn't negate what the student has said. Then we've got 23 to 25. So 21, 22, 23. The bases are joined together in pairs, a single base from one chain being hydrogen bonded to a single base from the other. Again, this doesn't negate what the student has said. So the answer must be D. Okay, but I'm going to go through D. I'm going to read the lines. So 27 to 29. Always make sure of your right answer. And you need to know why the other answers are wrong. Okay, you need to know why the other answers are not the right answer. By doing this, you can actually have a higher probability of getting the right answer. So one member of a pair must be a purine and the other a pyrimidine in order to bridge between the two chains. So now this negates what the student has said. He said they could pair up randomly. Now the text says no. It says a purine has to be paired with a pyrimidine in order to bridge between the two chains. So that's why D is the right answer. In the second paragraph, lines 12 to 19, what do the authors claim to be a feature of biological interest? So 12 to 19, okay? So the first feature of our structure, which is of biological interest, is that it consists not of one chain, but of two. Go back down. So the chemical formula of DNA, not exactly. Okay, I could already see the right answer, which is DNA consisting of two chains, of course. Neither is this or this. So it's definitely D. So this shouldn't take a lot of time. Okay? The author's main purpose of including the information about X-ray evidence and density is to, let's go back again to see why this evidence was used. So it has often been assumed that since there was only one chain in the chemical formula, there would be only one in the structural unit. However, the density taken with the X-ray evidence suggests very strongly that there are two chains. So they used the X-ray evidence to show that there are actually two chains within the DNA molecule, okay, or whatever. So establish that DNA is the molecule that carries the genetic information. Not exactly, again. They use the X-ray evidence to suggest that there are two chains. Presents an alternate hypothesis about the composition of a nucleotide? Not exactly. Provides support for the author's claim about the number of chains in a molecule of DNA? Well, yes. Okay. So this supports the author's claim that there are 2 chains in a molecule of DNA. So C is our right answer. Confirm the relationship between the density of DNA and the known chemical formula of DNA? Not really. Again, none of this was mentioned in the text, but C was. Okay. Now, based on the passage, the author's statement, if a pair consisted of 2 purines, for example, there would be no room for it, no room for the pair, implies that a pair of purines would be larger than the space between a sugar and a phosphate group, or of purines would be larger than a pair consisting of a purine and a pyrimidine, of pyrimidines would be larger than a pair of purines, consisting of a purine and a pyrimidine would be larger than a pair of pyrimidines. Okay, so lots of names here, lots of confusion. So again, if you go back to this section, okay, 29 to 30. So one member of a pair must be a purine and the other a pyrimidine. If a pair consisted of 2 purines, there would be no room for it. So again, this implies that a pair of purines would be larger than the space between a sugar and the phosphate group. Not exactly, okay? Remember, the bond, okay, needs to be between a purine and the pyrimidine. Okay, we're not talking about the space between a sugar and a phosphate group. We're talking about the space that extends between a purine and a pyrimidine. So this implies that a pair of purines would be larger than a pair consisting of a purine and a pyrimidine. Well, that's true, because again, we're trying the combination of a purine and a pyrimidine gives us the space that we need, okay, between the 2 chains. So this is actually correct. But when you have 2 purines or 2 pyrimidines, there isn't enough room for them. Okay, that's how they complement each other. A pair of pyrimidines would be larger than a pair of purines. Well, we don't have evidence for this in the text, and neither do we have evidence for this in the text. So consisting of a purine and a pyrimidine would be larger than a pair of pyrimidines. Well, not exactly. If you have a pair of a purine and a pyrimidine, you have the exactly the space enough to form the combination or the bonds. Now, the author's use of the words exact, okay, specific, and complement in lines 47 to 49, the final paragraph, functions mainly too. So let's go back to lines 47 to 49. If the actual order of the bases on one of the pair of chains were given, one could write down the exact order, so here we have the word exact, of the bases on the other one, because of the specific pairing. Thus, one chain, as it were, is the complement of the other, and it is this feature which suggests how the deoxyribonucleic acid molecule might duplicate itself. So let's go back down. So this information suggests that, okay, mainly functions to do what? Confirm that the nucleotide sequences are known for most molecules of DNA. Well, again, this is not what the text said. The text says since we know the exact specific pairing or the pattern of pairing, which is again adenine and thymine and guanine and cytosine, since we know this specific pattern, then if we have one chain, we could immediately deduce what's on the other chain. And this, of course, it tells us about how the DNA might duplicate itself. This is the information suggested by the passage. It functions mainly to counter the claim that the sequences of bases along a chain can occur in any order. Again, this is not what this specific lines are saying. Okay, now the text does say that the certain bases can be in any order. We can have any number of permutations as long as there's the opposite pairing on the other side. But this is not the main claim of lines 47 to 49. I support the claim that the phosphate sugar backbone of the author's model is completely irregular. Again, they didn't talk about the regularity of the author's model. So again, D has to be the answer, but let's see why. Emphasize how one chain of DNA may serve as a template to be copied during DNA replication. Well, yes. If we have one chain of DNA, this could help us to copy the other chain, okay, during DNA replication. Okay, we can have the copy of the other chain just by knowing one of them. So D is fine. Now, based on the table and passage, which choice gives the correct percentages of the purines in yeast DNA? Now, as we said, the purines are adenine and guanine, okay? And they are asking about yeast, okay? So the percentages for yeast are 31.3 and 18.7. So going back down, again, they asked about yeast. So 17, hold on. Okay, I forgot my numbers. So 31.3 and 18.7. 31.3 and 18.7. There it is. That's C. Okay. So, do the data in the table support the author's proposed pairing of bases in DNA? Do they support the author's claim, okay, or the author's suggestion concerning how the nitrogenous bases, the 4 nitrogenous bases, the way that they are paired, okay? So, again, let's go back up and let's take a look at this, okay? So, adenine, take a look at this. Now, adenine and thymine, if they are found in almost exactly the same amount, almost, okay? And this is repeating itself again in almost every single one of the organisms that we have. So 26.8 is very close to 27.2, and 33.2 is very similar to 31.6, 28, 28.4, 28.6, 28.4, 29.3, 30, and so on. Now, similarly, okay, if we take a look at the others, okay, so 22.8 is very similar to 23.2, 17.6, 17.6, 22, 21.6. So these amounts are really close to each other, which indicates that we have the same amount of those pairs, which indicates that they are, in a sense, pairing with each other. Okay, but I mean, if, let's say, just for the sake of argument, that adenine is going to join with guanine, then you have a 4% more adenine than guanine. Well, what's that percentage going to do? Okay, so again, let's take a look at what the options have. So, do the data in the table support the author's proposed pairing of bases in DNA? Well, yes, it does. Okay, and for one main reason, and that's that they are found in almost the same amounts. Okay, so yes, because for each given organism, the percentage of adenine is closest to the percentage of thymine, and the percentage of guanine is closest to the percentage of cytosine. Again, that is true. Okay, so adenine is very similar to thymine, and guanine is very similar to cytosine in amounts, in percentage of amount. So actually, I'm not going to read the rest because, I mean, immediately you could cancel no. So you're stuck between those two. Okay, now you only read for Y, I read A, I have my correct reason, so there is no need for me to read B. Now, according to the table, which of the following pairs of base percentages in sea urchin DNA provides evidence in support of the answer to the previous question? Okay, so again, in the previous question, we said that the base pairs, okay, they exist in almost the same amount or the same percentages. So which which following pairs of base percentages in sea urchin DNA provides evidence in support of the answer to the previous question? Let's take a look at the sea urchin, okay? Here's the sea urchin. Now, I want numbers in my options that are 32.8 and 32.1 or 17.7 and 17.3. If I have one of those, and I will have probably, okay, then I'm fine. I'll have my correct answer, of course. So going back down, again, 17.7, 17.3, or 32.8, 32.1. So 17.3, 17.7, here's my answer, and it's A. As for the rest, I mean, it's quite clear that the other options don't actually answer the question correctly because the percentages are so different from each other. Now, based on the table, is the percentage of adenine in each organism's DNA the same, or does it vary? And which statement made by the author is most consistent with the data? Okay, again, based on the table, is the percentage of adenine in each organism's DNA the same, or does it vary? And which statement made by the author is most consistent with that data? Of course, we could see that the percentage of adenine is actually changing in each and every organism. And it ranges from, let's say, we've got the lowest percentage of adenine is 24.7 in E. coli, and the highest amount is probably in the octopus, which is 33.2. So basically, the percentages are changing. Okay, so, and do they go in line? Or does this data agree with what the author had to say? Okay, so it varies. So we're gonna cancel A and B. it varies. Now, for which reason? We need to take a look at these lines, 36 to 38. So adenine, for example, can occur on either chain, but when it does, its partner on the other chain must always be thymine. So now this has nothing to do with the data that's found in the diagram. So the answer is probably going to be D, but I'm going to check and see why. Lines 41 to 45. I'm just going to make sure. So 41 to 45. Here they said it follows that in a long molecule, many different permutations, okay, are possible. So you could have any kind of repetition, okay. So you could have a lot of adenine, okay, or a lot of guanine or whatever, but they could be in any order, okay, along as, again, the pairs of nitrogenous bases are there, then you're fine. But you could have any different number of permutations. It's possible, okay. So D is our answer. So that's it. That's the third passage of the first practice test on the College Board website. The sample practice exams, you could go on their website and download them for free. If you have any questions, make sure you put them down in the comments section. Make sure you hit that like button, subscribe button. Make sure you share these videos with your friends and family. Thank you a lot for watching. See you soon in the next video. Bye.